Hello, everybody. Welcome back to Couch of Science. My name is Michele. I will be your host for tonight. And today we have a very nice topic for this new edition of Couch of Science. First of all, before we start, just a little reminder. First of all, sorry for the delay in the beginning, but we had a small issue with a technical issue, but we were able to fix it. And today we're going to talk about how do we survive in the space. And we're going to do with a person that is studying this topic. He has a huge experience, and we are going to learn a lot with her. But before that, I'm going to introduce another volunteer of Pint of Science in this Couch of Science night. And she's going to be the co-host of tonight, and she's going to talk about an introduction of what we are going to do in this wonderful night. Tanin.
Hey, how's it going, guys?
All good. And you, Tanin?
It's going well. So indeed, I am going to give a quick introduction just about the extreme things our bodies can handle. So very general introduction.
Space is very scary. Space is very extreme.
Yeah.
We are going, of course, to discuss this with our guest much more. But actually, you prepare something for us. And without wasting more time, I will just let you the stage. And so how extreme can be our body in a certain way?
Yeah. So it's first of all, like, what are you looking at? What kind of question you are asking? Are you looking at the personal limitations of the body, or if you put the body in the environment, what are some of the environmental extremes the body can withstand? So there are a lot of studies out there that go into all of these different categories, which I've summarized here on this slide. So very quickly, I think just because our speaker tonight will talk about what it's like to be in the environmental extreme, instead we're going to focus on the environmental aspects. So some things that I think that you guys should think about. So what are some of the physical limitations of the human body when exposed to different environments? What do you know of? Just bring up what general knowledge you have. And also how not to study the limitations of the human body. This has been quite important in a historical context. And there are some cool people who beat the odds every day. So what are the physical limitations of the human body? So we are going to focus on the environmental one. And what's more environmental than the temperature? So for example, the coldest temperature that the body can withstand is 21 degrees, but typically they die shortly afterwards. And when I mean the body itself, I'm talking about the core temperature of the body, not the temperature that you feel in your hands and your feet. Because when you start reducing your temperature of your body, your general organ system, all of the fine-tuned mechanisms within your body begin to slowly shut down. So there are some telltale signs when your body temperature is becoming lower and lower. Some of this research has been done in the military, in the Thermal Mountain Medicine Division of the US Army. They demonstrate that you can first start losing your memory, which is amnesia, then you lose consciousness, which is you basically black out, and then you die. So that's basically try to keep it above 33 degrees if you can in your internal body temperature. Next, in terms of extreme temperature of extreme heat, we have two types of reactions in the body. So you have heat exhaustion versus heat stroke. So in heat exhaustion, your body is exposed to general heat, you have responses, you try to sweat to keep yourself cool, your body starts to react in ways that are not life-threatening. You just need to try to drink more water, get into a cooler environment if you can. But heat stroke, on the other hand, is when your body temperature rises. So this is an American graph, so this is in Fahrenheit. Don't worry, that's not 103 degrees centigrade. It's actually 40 degrees centigrade, so the risk is a lot closer than we fear. So that is usually associated with a throbbing headache when you stop sweating because you've completely depleted your resources. You start to vomit, and you also have something called tachycardia, which is a rapid, strong pulse. and you start losing consciousness. This is the point when you need to go to a hospital and they need to take care of you because you may die. So that is the extreme limit that your body can withstand heat. So another environmental component is pressure and altitude. So for example, if you decide to climb a mountain, like some people do in the summertime, or in the winter if you're crazy, there is something that you should be aware of, is that as you go up further in the atmosphere, in higher altitudes, the pressure is decreasing, which means that the particles spread out a bit more and therefore are not readily available to you. So there is an image on the right of a hot air balloon, and as you can see, the higher the altitude, which is denoted by the numbers, the larger the balloon gets, because there's an expansion of the molecules that are inside the balloon. So that means we have an expansion of oxygen, which we need to breathe, and therefore it's more difficult for us to breathe, and we get something called altitude sickness. So what is the maximum altitude that we can comfortably maybe live on? Is 2,500 meters. There are people who were living for 2 years and then they came down because I guess altitude sickness got to be too much. But if you exceed 8,000 meters without any oxygen tanks, you're in the death zone. So try not to climb too high without some assistance. Another aspect of the pressure and altitude is when you go scuba diving or deep sea diving. So again, same principle, but reverse. So you have increased pressure buildup in your arteries, and the blood particles within cells, they start creating gaseous bubbles if you ascend too quickly, because you have a response to the change in the pressure. That's why if you decide to get a deep sea diving certificate, you have to dive deeply with a pressurized suit and a pressurized oxygen tank, and remember to slowly come up. Otherwise, you can even become paralyzed. So most recreational divers, they use a planner to determine what is safe and what is not. So these are some of the extreme limitations that your body can handle. And unfortunately, some of this information was determined with the trials during World War II conducted by the Nazis and concentration camps. So this is actually not 100% useful. They only got tidbits of information, and I'll get into why. So for some of the research that I will say was conducted was testing the limitations of the human body in terms of high altitude, freezing temperature, phosgene exposure, which is a poisonous gas, forcing people to drink poison and forcing people to drink seawater in case German soldiers or Nazi soldiers were exposed to unpleasant conditions. And how they did this was they unfortunately rounded up people in the concentration camps, and they would eject them in high altitudes, and they would perform brain surgery to see what the effects were, and they would basically torture people in weird sadistic ways. And why isn't it good research besides the major ethical dilemma is because the way that the research was conducted was not correct enough, in the sense that it was biased, that there were some weird eugenics in the background for some explanation of what a human can withstand. And on top of it, there was a lot of missing information, so there was no complete picture or a well-designed experiment. So unfortunately, there is this large debate as to what is ethical from these types of research. Like, can we use this specific information? So World War II ended, and these atrocities were brought to the justice system, and something called the Nuremberg Military Tribunal was created in 1947, where they put these doctors on trials, and they came up with something called the Nuremberg Code, which is now implemented in a lot of governments, because this is how to ethically conduct research. And some of the major points are explained below, that before you check to see what the extreme limitations of the human body is, you need to make sure you're conducting it in a safe way. So again, voluntary consent. They tried to, unfortunately, do animal experiments beforehand, and there has to be a benefit for the participant in conducting the experiment. So unfortunately, after the Nuremberg Code was passed, there still was some weird experiments in the loophole. For this one is part of, like, I think the top 30 horrible experiments conducted in human history. This was conducted by Karl Heller, an endocrinologist based in the United States. Essentially, he wanted to see what it was like to irradiate testes, because testes have cells that continuously regenerate. They undergo specific cell cycles, so we thought it would be interesting to see what the radiation was like. And he downplayed the risks for the prisoners, and unfortunately, there was some awful reactions to the experiments. But the results were still somewhat interesting for future scientists. Apparently, NASA was quite interested when they were sending astronauts into space, which is quite related to the topic that will be coming up. But there are some weird experiments out there. It's just a Google away, and it's pretty fascinating and horrifying at the same time. But do not be deterred by these limitations that I've described for you. There are daredevils out there like Felix Baumgartner that said, high altitude, nothing to me. Let me just jump off the stratosphere, which is, I think, about 39 kilometers into the sky, and free fall for 11 minutes, breaking the sound barrier and setting a world record for the highest jump. So there are people who like to test these limitations that our feeble human bodies are subjected to. So that's essentially the summary. So I'm excited to learn how to protect astronauts in space from radiation exposure.
Thank you so much, Tanin. It was a very nice presentation, a bit scary, but anyway, fascinating on the fact that how we push our body in the limits like Felix, or how cruel can be people pushing others to their limits.
What's happening?
Yeah, it's quite scary. It's quite scary. So before to introduce our guest, I just want to remember to everybody that is here for us and for her that you can interact with us through the comment section in Facebook or YouTube, depends on where you are following us. If you have any questions, just drop a questions there. At the end of the presentation of our guest, there will be also a Q&A session. And of course, if you have a beer, grab a beer, enjoy the beer, and find a cozy couch where you can sit, and so you can enjoy this nice talk. So without other interruption, I just want to introduce our guest. She's the director of Radiology Unit at the Belgian Nuclear Research Centre, guest professor at Ghent University and KULeuven University for Radiation Biology, Radiation Protection, and Space Biology and Medicine. Please welcome, and we are very honored to have her here, Professor Sarah Baatout. Hello, Professor. Welcome.
Hello, Michele, and hello, Tanin. Thank you very much. Thank you very much for this very nice introduction, and straight into the subject of astronauts' health. In fact, very related with all these extreme stressors that you have been mentioning, and we'll go back to these some of these stressors today during the presentation.
Okay, very, very nice. So first of all, thank you to be with us, and I hope everything is fine. And again, I hope that the technical problem is not going to interfere with the presentation, but we have everything under control for now. So I will just project the presentation, and I will just be on your service to change the slide when is the time. Okay?
Thank you again. Thank you.
And let's see. Okay, perfect. So, Professor, the stage is yours. Go on, okay. And let's see.
Thank you. So thank you, both of you, and thank you all for joining this couch of science. It's really a nice way to put and to join science and pleasure of the evening. And I hope that all of you are safe enough, and despite the very difficult situation that we are all encountering with this nasty virus. So today I will speak about how do we protect astronauts from space radiation. We already had some very nice introduction about radiation from Tanin, and we'll go more into detail. Could you pass the next slide? So I'm working at the Belgian Nuclear Research Centre. The Belgian Nuclear Research Centre is a federal institute of research. It's located in the north of the country, not that far from the Netherlands. If you do make a triangle between Leuven and Antwerp, you will find the third corner of the triangle angle of the triangle will be where MOL is situated. So MOL is an institute of research, and we conduct quite a lot of space research there. Not only, but quite some efficient and very important research for long-term space mission. And today I will really speak more about human space exploration, putting the accent on human, on the word human. So really going back to the introduction of Tanin about how the human body reacts in an extreme environment, because space can be regarded as an extreme environment. And the idea of the research that we are performing is really to support a safe, a productive, a healthy human space exploration of the solar system. How can we do that, and how can we contribute? Mainly from different aspects. One is to be able to characterize the type of radiation, and we will see that it's not an easy task to characterize, to monitor, to measure radiation, cosmic radiation in space, and that it's quite variable, and it's quite important to develop this kind of expertise. So for that, we have a very strong group of researchers, dosimetrists, they are physicists, and they measure radiation through different types of instruments inside and outside space vehicles. Then on top of that, we also have a research group really related to microbiology. And we will see in the next slides the importance of microbes, not only the microbes in our body, our microbiome, inside our gut, for example, but also the microbes that are inside a space vehicle, how they can interact and interfere with the health of the astronauts. And then we have the radiobiologists, the biologists really that are studying the impact of radiation and space radiation on health, and how can we better protect astronauts from space radiation. And on top of that, we have really a long expertise in studying the resistance of any kind of material to radiation. And we also develop different type of research related to radioactive sources for space propulsion. Next slide, please. We are really looking now, the slide before, if possible. Yes, so thank you very much. So we are now, if you look at in terms of time and timeline, we are now about 59 years of space missions. We have an expertise of about 59 years from the first orbit, from Yuri Gagarin in 1961, through the different types of programs from Skylab, the Mir station. We are now very much using the ISS, the International Space Station, where astronauts are mainly staying about 6 months, between 6 months and 1 year. But what is ahead of us is really going forward and having and developing a moon village, and thereafter also to go to Mars. And what we have collected, Tanin explained a little bit about what we already know from certain extreme environments that are not space related, but other extreme environments that help to decipher and to understand how the body cope and how long the body can cope to certain extreme conditions. So we know more and more about what is the impact, but we can expect even more impact the longer the duration of the missions. Next slide, please. What is in front of us? The endeavor, the big endeavor in front of us is really going back to the moon, having a settlement there, a moon village, and then the next step is really going forward to Mars. And this whole program is called the Deep Space Gateway. And the Deep Space Gateway would be the first human spaceship to explore the solar system, would be the most distant human space missions that would have ever been attempted, and would be the testing ground for understanding all the stressors, the challenges that are coming from long-term human space exploration missions in the deep space. But there is an issue, a big concern and a big issue. It's the ionizing radiation. It's cosmic radiation. Next slide, please. How can we really cope against that? And it's really regarded, radiation, space radiation is really regarded as the #1 risk to astronaut health beyond a low Earth orbit. So it's really a concern where we need to find what we called in space jargon mitigation. So countermeasures, ways to be able to counteract the impact of radiation. And I will speak about how we can do that. Next slide, please. On Earth, we are very well protected. We are cocooned. We are protected by our atmosphere, by our magnetic field that really stop about 99.9% of space radiation. So basically, a very small portion is reaching us on Earth. So no issue of radiation really, except if you go under the sun for many hours without protecting yourself, then it can be an issue, but it's more non-ionizing radiation. But ionizing radiation are not such a problematic. Next slide, please. Now, this is an important picture because it tells you and it shows you what is the increase of radiation we can expect from space missions. And if you look at the bar corresponding to 1 millisievert of a dose, this is the dose that we receive, so the residual dose that we receive on Earth per year. This is what the population would receive from the environment, not from medical applications, but just from the environment, from the solar system, from cosmic radiation, and also from the crust, the Earth crust, from radon exposure. So this is about 1 millisievert. When we speak about going back to the moon, having a moon settlement, these are longer missions where astronauts would stay for about 6 months. Then we see that we increase of a factor of 300 the dose of radiation in comparison with what we have on Earth. And then if you look on this graph for Mars, we are even increasing of a factor of 1,000 for the whole mission to Mars and back. We have about 1,000 more radiation to face. Next slide, please. So I have to explain you and to define a little bit what are these space radiations? What are they composed of? I mentioned that they are quite complex to measure. They can be divided into 3 kinds of categories. First, the solar wind, which is a constant flux of low energy particles. It's continuous, but for us, it's not such a big issue. It represents a low hazard, and it can be stopped. It can be blocked with the space vehicle, with the armature and the coat of the space vehicle. So for us, that's not such a big issue. What is more of a concern are the solar particle events. It's the solar flares. It's when the sun is extremely active and starts eruptions on the surface of the sun. And there, at each of these eruptions, at each of these solar flares, you can have a huge flux of high energy protons that can last for a day or 2, but it's extremely hazardous. It's the astronauts that really have got to protect themselves, and we will see how do they do that on board of the International Space Station in a few minutes. Where we don't have a solution and where we really need to continue and pursue into this research is for galactic cosmic radiations. For galactic cosmic radiations that are also a continuous flux of high energy protons, of heavy ions and alpha particles, so very damaging radiations that can really induce a lot of complex damage inside the cells, where we have some kind of start of solutions, but where we still need to continue research. So this is just to define a little bit what space radiations are. If I may go to the next slide, I just would like to emphasize now what how important the International Space Station is for researchers like me and like my team, and for many space researchers all through Europe, as well as in the United States and in Russia and in Canada and Japan, because the ISS is used by all these countries. The ISS, the International Space Station, is in orbit at 300, 400 kilometers around the Earth. And that's where a lot of research is conducted. It's really the starting point, and the preparation is taking place for long-term missions to the Moon and to Mars. For the moment, we have radiation detectors. Our physicists are sending every 6 months new types and new detectors to measure, to monitor radiation very precisely inside the ISS. We also conduct, my lab conduct different studies on health impact of astronauts. So for that, we measure before the astronauts are leaving the Earth and before they go on the ISS, we measure their radiation sensitivity, how susceptible are they to radiation, and we measure them also when they come back. So we conduct human health monitoring. And but we're also very involved in what we call the MELISA program. And the MELISA program is the program of the European Space Agency for life support, in order to guarantee astronauts to survive to on the Moon, and to Mars, on the way to Mars and back. We need to provide them some food, and we will see how important it is to rely on microbes, on bacteria, in order to purify waste, purify feces, urine, and into water and into fertilizer for plants. Next slide, please. And just a slide here as an introduction to our dosimetry. You see on the picture, there is a little orange box, and this is one of our detector inside the International Space Station that allows us to measure the total quantity of radiation that can reach inside the International Space Station. Next slide, please. On this slide, you might recognize one of my hero, one of our big name here in Europe, in Belgium as well. It's Frank de Winne. Frank de Winne is our second astronaut, Belgian astronaut. And here is attaching inside the Columbus module. The Columbus module is something we can be very proud of. It's the ESA, the European Scientific Module. And inside this module, you see all the little red squares distributed nicely inside the Columbus module that represent each of them one of the detectors in order to measure radiation. So we are very lucky to be able to rely on the support of Frank de Winne, who is now head of the European Astronaut Center in Cologne. Next slide, please. This is a map of the International Space Station, but just a flat map, where you see the three parts that are shown in red are the places and where the astronauts can go in case of solar flares. So in case of very high flux of irradiation, they can rely and be protected in this one of these three compartments, where they will stay all the time when a solar flare is hitting the ISS. These solar particle events, these solar flares, are detected just minutes before the solar flare arrives and reaches the International Space Station, and they just have a few minutes then to go and to protect themselves inside one of these three parts. Next slide, please. But on the Moon, just here a picture of a possible moon village. It's really like a universal endeavor. It's not only a European project, but it's also a NASA project, an international moon village. Russians are also involved, Japanese, the Canadians, and moon village would be a place where humans and robots interact and they work together and develop and perform different types of studies, research, and projects on the lunar surface. Very important to go back to the Moon to understand the Earth history. But there on the moon there won't be any magnetosphere. There is no atmosphere. So there is no protection from the harmful space radiation. So we need really to find the best way to protect. And the moon is considered as the first step before Mars. So to test, as a testing platform, to test all the infrastructure, the equipment before going and heading towards Mars. Next slide, please. On the moon surface, inside the moon village, astronauts will have to face and will go out of this moon village quite a certain number of times. What we are used to historically from the Apollo program is maximum about 20 EVA that were performed during the whole Apollo program. And the record right now is 3 lunar extravehicular activities during this Apollo program for an astronaut. But in the future, in the moon village, these extravehicular activities, so when the astronaut is going out and go for exploration on the surface, go take some samples on the moon surface, do some types of experiments, it's foreseen to have about 70-80 different missions per astronaut outside the moon village, which means that in those situations, the spacesuit will be the only and the solely protection of the astronaut. So it's very important to develop the most and the best spacesuit we can think of in order to protect, especially against radiation. Next slide, please. And my institute, the Belgian Nuclear Research Centre, is also working on the best composition of the spacesuit, because the spacesuit will need to be resistant against abrasion of the reactive, particularly aggressive chemical dust, resistant against the UV, resistant against temperature, being able to keep oxygen, and also have a kind of light weight. So, and it also needs to be flexible enough and elastic enough to allow the astronaut to go again and again outside, inside, and walking and moving around, sampling. So it's important to perform research into the best composition of the space suit. Next slide, please. Radiation on Mars. The next step will be about 1,000 times radiation dose in total from what we have on Earth. We know that Mars is the planet which is the most similar to the Earth. It's very important also as a fundamental question for humanity to understand whether life existed elsewhere in the universe beyond Earth, because we know that Mars was once full of water and warmer than it is now. It has a little atmosphere, 1% of atmosphere, so it's potentially a kind of habitable environment. And so that will be the next step, but radiation is, of course, a main issue. Next slide, please. A little bit of medicine there. We know that radiation represents a high risk for health, because it can increase the risk of developing cancer. This is what we call carcinogenesis, of initiating cancer. It can also increase impact and effect on the central nervous system, on the brain, with some changes that can be induced in cognition, so, and in memory, in mood, in behavior, in changing the behavior of astronauts, in motor function. But it can also increase the risk of cardiovascular diseases, amongst others, stroke or lens opacity, cataract. And also space is recognized as also an accelerated aging inducer. So there are quite a few things to take into account, because most of the systems of the body are quite sensitive to radiation. And also in terms of high doses of radiation, when we have a solar flare of very high dose, you can develop an acute radiation syndrome. If you have seen the series Chernobyl, you have seen some people who developed this acute radiation syndrome with nausea, vomiting, fatigue, extreme fatigue, skin reddening, depletion of the bone marrow and of the blood forming organs. So these are really high risk. We can't really take because we have to be careful enough. So we really have to follow the program and the procedure to limit as much as possible the radiation dose to astronauts. Also a complexity, an extra layer of complexity, if I may have the next slide, is the way space radiation hit the cells inside our body. And you can see on the right hand side some pictures where in blue are represented the cells, and then in green you see the different spots that are really cutting in a way the cells, like cutting with a knife a piece of meat. And so basically when the DNA is cut, all these green spots inside the nucleus are really representing broken DNA, a broken DNA. And the more and the closer all these dots are, these green dots, the higher, the more difficult it is for the cell to repair. And it's the higher the risk that the repair is not done properly. So there are really a lot of questions still to answer to in order to understand what is the unique damage that is induced to the biomolecules inside the cells, to the tissues, to the cells. How can we distinguish the DNA damage patterns between cosmic radiation to what we know on Earth, ionizing radiation from Earth, what we know from conventional radiotherapy, for example. So there is really a lot of research still needed on the cellular point of view, but also on animal models and also on astronauts. Next slide, please. When we look at the way the body reacts to radiation, we can see that not everyone is equal in front of radiation. So some of us are very sensitive to radiation, some of us are very resistant to radiation. And as we can see on this picture, we have a Gaussian curve. The majority of people have got a kind of a moderate, normal radiation sensitivity, but we can't exclude very sensitive people or very resistant people. So it's important, as we do in radiotherapy context, in radiotherapy research, develop precision medicine in order to adapt a radiotherapy treatment for each patient. We do the same for astronauts, developing precision medicine, developing programs for each of the astronauts to go to the moon and to go to Mars, the best program fitting with their radiation sensitivity, allowing a certain number of times they could go outside the moon village, for example, depending on their susceptibility to radiation. Next slide, please. Also, radiation sensitivity changes with age. The younger, the higher the sensitivity. So if you clearly look here on the right hand side, you see that in fetus, in very young children, you have a very high sensitivity. So that's why also children, we try to of course avoid irradiation, also as much as possible, medical irradiation and also radiotherapy if possible, of course. But if the benefit is great, if you want to cure cancer, radiation is sometimes the best option that you can get. But radiation sensitivity varies with age and decreases with age. So, which means also for space programs to go to the moon, to Mars, it's important to take age as one of the determining impact factor. And as you can see, from the age of 50, radiation sensitivity is much lower. So it might be best to take on board for long-term missions 50 years old onwards astronauts. Next slide, please. So also to show that radiation varies depending on the gender. You see here a picture showing what we call in our jargon, sorry for the wording, but it's the risk of radiation-induced death. It just shows you that basically females are more sensitive than males. And if you take into account that radiation varies depending on the age, depending on the gender, depending on the genetic susceptibility, so it's really important to have some good precision medicine and to have some good harmonization into the program of radiation protection of astronauts. Next slide, please. So back to precision medicine in astronauts. How do we do that? How can we develop the best program of radiation exposure per astronauts? For that, we need biomarkers. We need to understand and develop better biomarkers. And biomarkers are any type of molecules that could reflect the interaction between the biological molecules, so inside the cells, the DNA, for example, of an astronaut, and the environment, so the space environment, for example, and which needs to be very precise and specific to the space environment. Next slide, please. So this is one of the type of research that we are currently doing at the Belgian Nuclear Research Centre, where we get blood from the astronauts. This is one of the crew that we've been monitoring. Maybe you can recognize Scott Kelly there. And in order to, that's very helpful and very important to be able to monitor radiation sensitivity all through the mission, and also before, to understand which astronauts is particularly sensitive to radiation, which is less sensitive to radiation. Next slide, please. So understanding radiation tissue sensitivity is really crucial. Here, another experiment that we flew to through the Russian Space Agency. On the left-hand side, you see the photon M3, and this was a skin experiment, because skin is aging extremely fast in space conditions. And you can see the ground samples on the left-hand side, and on the right-hand side, the space samples, where if you look at the colors, focus please on the green and on the red on the pictures, you will see that basically the damage inside the space samples of skin was much higher than the control samples that stay on the Earth. Next slide. One of the particular issue that we also need to solve is the vasculature, the blood vessels. We have blood vessels everywhere, of course, in our body. And these blood vessels are extremely crucial. They help us to irrigate the body, to bring the blood wherever it needs to be, and in all the parts of the body. And these blood vessels are quite sensitive to space conditions, not only to radiation, but also to microgravity, to weightlessness. So it's very important to understand how we can better protect blood vessels, because if blood vessels starts to explode, you will have an hemorrhage, and that's it's very difficult to cure that and to treat that in space. So it's very important also to protect the vasculature of the blood vessels. And this is one of the experiments that we flew with SpaceX, where we grew blood vessels in space. Next slide, please. The brain, as I mentioned earlier, the brain is also can be also altered, impaired in space conditions. So also important to understand what is the specific tissue sensitivity of the brain. And you can see on at the bottom on the left hand side some brain cells coming from a mouse brain. In the middle, you can see when they are exposed to microgravity with the machine that you see on the top right, they develop a lot less well. Their network, the neuronal network, is not as well developed. So it really means that the number of neurons might decrease, which can be quite a major issue for long term. The longer the duration of the mission, the higher the risk. And on the right hand side, you see the neurons with when they have been irradiated with simulated cosmic radiation, and you see that there are less cells and they are not so developed, and the prolongations between the cells is not that developed. So really the combination of microgravity and radiation can be quite harmful for brain cells in space. Next slide, please. Another issue that we are currently studying is the microbiome. In our gut, we have about 1 kilo of bacteria. And if you count this number of bacteria, they correspond to about 10 times more cells than human cells. So we could consider our body as a giant microbiome, because there are 10 times more bacteria than human health. And so we really need to take into account how they will react. And we need to quite preserve as much as we can the composition of the microbiome in space, in the space environment, because we know more and more that if we disturb this microbiome, this gut microbiome, it can really induce diseases. And we know that it's really involved in anxiety, for example, depression, and different types of diabetes, different types of diseases where the microbiome plays an important role. So very important to see how we can preserve a good composition of the microbiome in space conditions, and through transplantation of the good microbes during flight, all through the flight maybe, and by trying to preserve the good composition. Next slide, please. Another question which is very important in terms of research is also how can we understand radiation resistance in organisms, and how can we, with this know-how, expertise, can we increase this resistance of humans if we go to Mars, and maybe later to other planets, to Jupiter, who knows, the doses of radiation will be even higher for longer duration missions. So we need to really understand what we can do in order to mitigate that, maybe on a genetic point of view. And for that, we are really lucky. We work together with the University of Namur, and we together send already some rotifers. The rotifers are shown here in pictures, and they really are extremely resistant towards radiation. And they really help us to understand what makes them so resistant, in order that we could use as information in order to make maybe humans more resistant. And our next experiment will fly in December with SpaceX. So check out and follow that. Next slide, please. Life support. So bacteria, as I mentioned a little bit before, the microbiome is one very important aspect, the gut microbiome, to preserve it, to preserve the good composition. But also microbes are very interesting to recycle waste, to recycle, you can see here the Melissa loop, where basically you have different types of compartments with bacteria that on which my institute is working, that really help to close the loop. So basically, from the waste of the crew, the feces, the urine, you can purify through different compartments with essential bacteria. You can fertilize some plants, and you can send that inside the astronaut's compartment. And with pure water, purified water, bringing new CO2 and bringing food, because bacteria can be also an important food support. So SCKC and the Belgian Nuclear Research Centre is working for many years and was one of the founding members of this MELISA program of the European Space Agency. Next slide. I just would like to say a few words. Tanin mentioned some analogues, some very extreme environmental conditions, and she mentioned altitude. I just would like to mention Antarctica. Antarctica is one of the best analogues you can think of for mimicking, for simulating in the future lunar and Martian habitats. For many aspects, it's a very good analog for mission, the simulation of the mission, the environment, monotonous, hostile, for the habitat, closed habitat, where you are confined, where you have a limited resupply of food, and also for the social interaction with a small group, confined, international crews, and with the same type of personality, a high level of solidarity, and this endeavor and this motivation of, and general inclination of pushing boundaries in psychologically. Next slide. I was very honored and very happy to be able to go to Antarctica myself to perform some research there. You can see the beautiful Belgian Princess Elizabeth Station, where I spent a certain number of weeks monitoring the health status of the workers there and of the scientists, and understanding the impact of an extreme environment on health. Next slide, please. So what can we do to improve radiation resistance of astronauts. We can perform and improve the shielding, physically the protection, the type of material that we will use. We can also perform some medical selection of the astronauts and maybe select the ones that are the most radiation resistant. We work also on different types of molecules that the astronauts could take before going out of the moon village on a mission, on an extravehicular mission. So the types of radioprotectors. Also developing biomarkers that will allow to monitor how is it too far already? Did the astronauts reach the dose and the type of damage that he should reach, and should we stop the astronauts going outside the moon village, for example? And we can also play on the metabolism of the astronauts in order to decrease the metabolism. That's what we call hibernation, to increase radiation resistance. And also, what is very important is to think about regenerative technologies, taking on board for long-term missions all types of tissues from the astronauts in order to regenerate a tissue and the organ if any organ would fail to work. And I would like to invite you, if you are interested in this matter of research, to look at and read the article which is mentioned here under Vive la radiorésistance. This is a free article online. Next slide, please. So let's not forget that space radiation is very related and has many aspects related to patients and the benefits to patients on Earth. And one of the benefits of my everyday research and space research is really to even develop better radiotherapy treatment of cancer, because cancer is really a number one killer. We are in a population which is aging, so the longer we live, also the higher the risk we have to develop cancer, which is an accumulation of mutations. So cancer is going to continue to increase because we live longer, we survive cancer. So when more people develop cancer and might develop more than one cancer during their lifetime. And so this type of research, space research, has got really a direct link to curing and developing better treatment for cancer. Not only for that, for other aspects of research, cardiovascular research, neurological research, osteoporosis research, and we can discuss that further during the Q&A session. Next slide. If you are interested in this subject, I would like to invite you also to follow the TED conference on space radiation on that matter. Next slide is the acknowledgements. I would like to thank at the Belgian Nuclear Research Centre quite a lot of people that you can see on the left-hand side, and also different types of partners. I'm very, very lucky to work with, and the European Space Agency, the Belgian Science Policy, and the International Polar Foundation. And with that, I would like to finalize with the last slide, and thank you very, very much for your attention. And I would really be very happy to answer to some of your questions if you have any.
Thank you so much, Professor Baatout. It was very, very interesting, very dense, a lot of information. And but it seems to be a very critical point for the space exploration. And I have to be honest, I was not even not aware, but I was not even thinking about it. This might be so impactful for the space exploration. So the radiation, the effect of the radiation on our body. So mainly because probably something that, especially in movies, you don't see so much. They don't spend so much time talking about this kind of issues for space. So since this is the best way, or actually the common way that we have to interact and to think about space exploration, sometimes it's very difficult to imagine that. And anyway, so for our audience, I already see that there are some questions. If you want, please, guys, this is my moment to storm us with questions, because our guests will be more than happy to reply. Tanin, do you have any questions? Because
personally, for questions, oh my God, where to begin? There was
yeah.
It was really a really cool talk, and I was also quite delighted, because my specialty is the gut microbiome, to hear that it's also being used in such a interesting context as space. And okay, but I think I'm going to go on a ramble, so I think I'm actually going to read the questions in the comments because I'm just so excited. Okay, so there is a question from Michael Müller. He said, thanks for your presentation. How are materials tested in the practical sense? Are you using animal models? I'm guessing, how do you test materials against radiation? What do you do in order to make that happen?
So that very interesting question. So there are different ways to test. The first way is really to understand how the material itself will react. So for that you look at what we call the radiation aging. So how much the material is going to age. So how is it going to break? How is it going to stay in the same type of structure, or is it going to break into smaller pieces, or not be as thick? Let me give you an example. If you leave, for example, plastic under the sun, under the UV, for a certain number of weeks or months, you will see that the plastic might degrade, might become harder, might not be as flexible. So you can measure different types of characteristics in a material that shows you how the material is aging. So this is one of the first things you can do. Then the next step is to look at how it will protect. If you have a good candidate, a good material, or a good mixture of material, because we're talking also about different types of material being combined together, you can put cells and see how it protects cells from radiation. So for that you will irradiate with different forms of irradiation. Some can simulate part of the space spectrum. We can't simulate everything on Earth, simulate the whole space radiation spectrum, but we can simulate part of the spectrum of radiation, and that's what we do. So you test some of the material for which you think are the best composition, have got the best composition, and you look at the DNA damage inside the cells. And so the more damage you have inside the cells, the least efficient is the material.
Mm-hmm.
The next step, if you have a very efficient material where you see that on the cells there is not much radiation that really reach the cells, so it means that the material that you are using is really blocking very well radiation, the next step is you try that on animals. One of the animal model that we use the most is the mouse. We can use also rats, but mouse are usually a very good model. And mice as well as rats have flown quite a lot in space. There are quite a lot of experiments that have been performed, so we know quite well also the behavior of mice and the behavior of rats. And that will really help us also to understand at the in vivo, at the level of the whole body, not just the cells that you have done in the first experiment, but really at the level of all the different organs inside an animals, what will be the impact. And then you study the impact, short-term as well as long-term impact on these animals. And this long-term impact, if you think about, for example, cancer or cardiovascular diseases, they will not develop in one day. They will take a certain number of weeks in animals to develop. So you need to really perform a long-term study of a certain number of days or weeks if you want to study, for example, behavior, memory, learning capacities in animals. You really have to do different types of tests at different time points after the exposure to radiation. Now, what is also very interesting is that we have good models for simulated microgravity, because this is also where the Belgian Nuclear Research Centre is unique, because we can study these two stresses together. We have different platforms that are very unique, where we can simulate microgravity and irradiate at the same time. So then you can see whether microgravity alone and radiation alone can induce a certain type of stress. And when you put them together, you can understand that 1 + 1 = 3, that you really have a synergetic effect, that you have really an added effect of the two stresses together. So you reinforce, you have even more damage that you will induce inside the cells and inside the body. And so when your material is tested and you are satisfied, you can then perform some studies, some start of the studies on human cells to understand what would happen in a human body, and maybe propose that for radio protection, put a patent on that, and then maybe develop with a company a type of coat, a type of suit, spacesuit. We are working with a technological designer on a spacesuit with the best material we can think of for protecting against radiation.
And what is the best material that you've come up with so far?
So the best material are material with the smallest atoms. So the best material would contain very small atoms like hydrogen, like helium, like boron. Boron. So they are really the smallest atoms. So molecules, large molecules, like for example some plastic that are long chains of carbon, where we have hydrogen on each side of each carbon. These are long chain. For example, polyethylene. These are the plastic. For example, plastic bottles. Plastic bottles are long chains of carbons and hydrogens with a very high concentration of hydrogens. And I showed you the picture of the ISS, the flat map of the ISS. One of the compartments is the sleeping quarter, and the sleeping quarter is protected with layers of these plastic films, of this plastic bottle film, layers around their sleeping sac. And these are really good protectants. And another component could be, if we could be water, H2O, because you have also a lot of hydrogen there. So molecules where you have a lot of hydrogen, or where you have a lot of helium, a lot of boron. So we're also working on boron, but mainly on long chains of carbons with hydrogens. These are really good material to protect.
Do you think you can make these spacesuits out of recycled plastic even, as a possibility?
You could. The only thing with recycled plastic is that you could not make something extremely strong with that. It would be very good as a kind of as a layer, as a layer, but to keep that, to take into account that for the resistance, to breakage and so on, it's not that good. So you really need a combination of materials that are strong enough that will keep the space vehicle or the spacesuit still elastic but very resistant, but that is not going to melt. You need a component which is going to be resistant to temperature as well. We are really talking, and plastic is not always the best. It could be the best for certain aspects of research and certain like the space vehicle, inside the space vehicle, but don't put it as a coat outside. It would not be strong enough. So for the spacesuit, it could be very interesting. Plastic could be very interesting, but not only. So together with a combination, indeed.
It would be possible or imaginable to have as a layer of the astronaut spacesuit like an hydrogel, because you say that water actually can be something that can protect. So an hydrogel would be probably much easier to handle than water per se. But I don't know if this might make sense. Probably.
Yeah, but in fact, it's very interesting. This can be certainly very interesting. The issue is we would need about really to protect completely just with water. We would need 10, 11 meters of water.
Oh, wow. Okay, so no.
So for a spacesuit, it would not be possible, but maybe and we need to produce this water. So this is also a bit of an issue as well. But indeed, an hydrogel could be, because the spacesuit that is being worked on would be a kind of second skin. So will be something very close to the skin, attached to the skin, full of monitors, of detectors for radiation, but also for temperature, for different types of monitoring of human health, of different types of parameters of health. A continuous monitoring would be performed with this type of second skin of spacesuit, but also for monitoring the mood even. So depending on the movements, really trying to really understand how stressed the astronaut is, and depending on that, through also artificial intelligence, to be able to tell when it's time to go back inside the moon village, to finish the mission, the extravehicular mission, to go back inside. So they are really, but indeed, an hydrogel with a composition with water would be certainly interesting. Also, you could imagine using feces as a layer. There are research,
Wow.
or a layer of plants, growing plants, that could be of interest, maybe not for the spacesuit, but for the space, the moon village, for example. So all kinds of possible ideas are, but really in combination. So, but as I mentioned, water you would need really meters of water to really stop all radiation. So water alone would not be possible.
Okay, perfect. So I think this is in the same idea. There is also a question from David. Are biological fibers and filaments better than synthetic? Was what we were just discussing right now, I guess. So in terms of to protect,
this is a very interesting. We are studying both, basically. We're studying both, and it's. I would not conclude completely, but I would say I think synthetic fibers might seem to be more better than the natural one right now, but I don't want to really to conclude, because still some research is needed on that. And we had put really a lot of hope on natural fibers, and now we're a little bit, we still need some research, but I would say I think synthetic for the moment are best combinations than natural.
okay, perfect. So we have a lot of questions actually. We have this from Ophélie. First, thank you, super amazing presentation. Thank you to Pint of Science Belgium, of course. Considering the high concentration of radiation on Mars, do we know if these conditions are compatible for life? Are some organisms on Earth, such as rotifers you have mentioned, able to survive in these conditions?
For sure, rotifers would survive bacteria. Some bacteria would certainly survive. So microorganisms and early life could survive on the Mars environment with not too much issue. Some really extremophiles could really already surviving on Earth in extreme environments of pressure, of temperature, so of the space environment. Some lichens, yeast, bacteria, fungi are put outside the International Space Station, in the space vacuum, fully exposed to radiation, and they survive. So definitely, I think that life would be able to survive. However, for humans, it's different, because to kill some type of bacteria, you need sometimes 20,000 more dose, 20,000 times more dose than to kill a human being. So humans are very sensitive in comparison with bacteria and with the early forms of life. So a lot of research is still needed to improve this radiation resistance. We are not completely there yet. I would say if we would go on the Mars environment without any protection, humans would certainly not survive. So we need to work very well on being able to monitor this radiation, to mitigate, to stop it as much as we can. And then when we reach a certain dose, which is the threshold maximal dose for a certain astronaut, to put this astronaut back in full protection against radiation. And this is what we really have to do. And it's a combination of genetics, of pharmacology, of shielding, of changing metabolism, and of biomarker studies. So it's really all these type of research is being performed now to guarantee to a safe and a productive mission to Mars for the future astronauts. The European Space Agency is going to recruit astronauts in a few months' time. So if you are interested in applying, if you are older than 18 years old, between 18 and 50 years old, please think about applying. And if you are interested, and just to mention, the European Space Agency will recruit very soon. We need new astronauts, younger astronauts also, for going to the moon and to Mars.
So actually, that's a great advertisement, because there's a question from Françoise Massot asking what is actually the interest, or the incentive to send people to Mars. I guess what is the point? What maybe will they do ultimately? Because you are advertising.
Of course, these missions, before we really have like a Mars habitat on the Mars. On the Mars surface, it's not for tomorrow, and we are really talking about 2040, I would say, to have a Mars habitat, but 2030, 2035 to go to Mars with the very first mission. Really to understand, to push the limits. Mankind has always been full of adventures. People who really push the boundaries of knowledge of mankind, of trying to understand what is how life is being developed, has developed on Mars, and how can we cope. I think it's really part of our human nature to really push forward science and research. And I think let's not forget that all the research which is performed to push and to bring astronauts to the moon and to Mars is very helpful on Earth. I think it's not said enough, but all the research performed on radiation protection is directly implementable on patients. So what we develop, the biomarkers, the monitoring, protecting the tissue, the healthy tissue from radiation, is something we can directly use in the clinics. Osteoporosis, I didn't speak about the bones that are really aging extremely fast in space conditions. All the treatment, the molecules, the exercise, the physical exercise that are performed by astronauts, this is directly implementable on patients on Earth, elderly patients who have to who really suffer from osteoporosis. For cardiovascular diseases, also a lot of major input from space research. So there is really, it's not only just putting people on Mars or on the moon, but it's really everything that comes along with that, which is an enormous advancement of research and of medicine, thanks to this research.
Really cool.
Yeah. In fact, before you were talking about in these slides of all the different approach to protect astronauts from radiation, and was shocking to see that mainly of them are all latest field of medicine and biotechnology, like gene therapies or radioprotectors, all fields that actually have a huge application on Earth for everybody. So this is always, again, as you say, push the limits, which in the end is the main point of humankind in a certain way. So try to push more and more and more. And so we have some other questions. I don't know, we can go probably for this question to go briefly back to the point of materials. And Nena, she's asking, in one of the slides you are mentioning there are some shielding rooms in the ISS for protection of astronauts during solar flares. Are they foreseen of heavy materials to enhance photoelectric effect for shielding purposes?
So as I mentioned, so one of the rooms is one of the room which is very much shielded is next to the water tank. It's really next to the water tank of the astronauts. So that's water, as I mentioned, H2O with a lot of hydrogen is a very good protectant. So that's one of the shielded part. And one of the other one is the sleeping quarter, where you have a lot of polyethylene, so a lot of layers of carbon, chains of carbons and hydrogen. So again, in those two aspects, it's mainly hydrogen rich components that are providing some very good protection for the astronauts.
Are there any small proposed improvements that could be added to these rooms? This is a question from Rosita Carvajal. Sorry if I'm mispronouncing your name. Deepest apologies. In the ISS station, for example?
So you can yes, so you can think of even adapting spacesuit. You can even maybe think about what is also very important is that depending on the type of material of the space shuttle, of the coat, you can have a production of secondary particles, so that one when the galactic cosmic rays are hitting somehow the space shuttle, the space station, you can have a production of secondary particles, so generating a myriad of particles inside the vehicle. And so it's also interesting to have these layers with very low atomic molecules to stop these myriads of secondary particles. So for that, you have different types of components that you could think of putting, maybe not everywhere on the International Space Station, but in some areas that need to be protected, like the sleeping quarter, for example, where if the astronauts have got to stay there for solar flares, they might have to stay for a day or two. So for some of the parts, you can think about even protecting and stopping better with different types of molecules. So indeed, we are working on that. It's not yet totally finalized. If the person is interested in contacting me, please do so, and we can further discuss on. I don't know the background of this person. Maybe this person is very much into these types of radiation protection, shielding, and maybe that's for anyone who is interested in even going deeper into the scientific discussion. Please do not hesitate to contact me further to my email address. I think it was in the last slide. Or in the first slide, but my name is very easy to find on internet anyway.
Well, that's really cool. So how about we take things back down to earth as a final question? We have one question from MJ Schreimer. Also, sorry again if I butcher your name. She says, "Hi, personally, my skin is very sensitive to sunlight and I get sunburned really easily. Would this, hypothetically, influence my chances at space travel, and if so, how?" So it should, radio sensitive.
So yeah, indeed. So if you are very sensitive, like many people who are very sensitive to sun rays, usually it has some impact on the way you will repair DNA damage and the speed of DNA repair and the quality of DNA repair. So I would say, without total certainty, but I would say that the chance to be taken, if we would go for a selection and a screening taking into account radiation sensitivity, but in Europe. And for the moment, it's not really on the agenda to select astronauts for their radiation sensitivity. What is on the agenda is to develop precision medicine. So it's not because you are radiation sensitive or very radiation sensitive that you can't be candidate astronaut. However, you might have an adapted program of exposure to radiation, and this is what we are working on with an ESA topical team specialized on personalized medicine for astronauts to adapt the program. So to not select based on a genetic point of view, but to adapt the program. So the number of time the astronauts would be allowed to leave the moon village, for example, and to go on extravehicular activities, or the type of mission. Maybe the astronauts would be allowed to go to the moon, but not to Mars. This kind of thing, but not to select for radiation sensitivity. We give the information, we test the astronauts, but we are we ethically we are not allowed to select for these criteria, because we might also, by selecting for something, by selecting for the most radiation resistant astronauts, you might also pre-select them for something else, for being more prone to maybe develop something else, another disease, more easily. So it's very dangerous also to select on genetic basis, and and it's not on the agenda of the European Space Agency.
So we cannot imagine to have in the future a system that selects astronauts also on the genetic of that person. Is
No,
okay.
not,
Yeah, so it's our duty to have the tools to protect them if they want to have the
Absolutely.
ambition.
Yeah,
Exactly.
yeah. About that,
Technical way of doing it.
Everything we can to guarantee that they will stay healthy, and not just survive, but really being able to work to enjoy the mission, and not just to survive, because we survival is not enough, of course. We really, and so there is, it's going forward, research is going forward very well, but there is still quite a lot of questions to answer to. But if I look for the past about 20 years, I've been working in the field of radiation protection of astronauts, and we move forward really a lot. Still, we are not there yet, but most probably, I would say in about 15 years, we should make it, and be ready with everything we need in order to make it a safe trip for astronauts to go to the Moon and Mars.
okay. I would conclude with this last question from Fabiana da Pieve. As a scientist, what is your sincere opinion? Do you think it will be more feasible to find a radioprotector to be injected or ingested, or do you think there is still a lot of room to improve shielding materials?
Both.
Both. Okay.
Hand in hand, absolutely both. With the shielding, we need both, and the shielding, and the molecules, and the pharmacological agents. Absolutely, no doubt that it's going to be both together.
But very, very quickly, the radioprotector as a molecule will be just help to repair the DNA. This is the main idea, I guess.
And to induce less DNA damage, so to repair faster, or to decrease the rate of oxidation, so to decrease the rate of damage, to really to have a boosted immune system. So it can really decrease the original amount of DNA damage, and it can stimulate and boost the repair, indeed. And there are quite a few molecules that are being tested, and hopefully they are, so for the moment they are tested at the level of cells, at the level of animals, and they are very interesting. And we hope that they will soon also be tested at the level of the clinics, on patients, and who knows, later on astronauts as well.
Okay, perfect. Thank you so much. Professor Baatout, was very, very nice to have you. This talk was really interesting. So I saw in the comments a lot of questions, some of them quite specific, and I insist of this person, if she or him won't contact you to have extra information. And sorry, but our time is running off, so that's why we cannot.
If there are any questions that I didn't have the time to answer to, you can still forward them to me, and I can answer to them, and give you, and send you back the answer if you are interested.
That would be great. Actually, that would be very, very nice. So we can create a post on our Facebook page, though, where everybody can have a look of it. So again, thank you to everybody. Thank you to you, Professor. Was really nice to have you, and thank you to our audience. Thank you to you, Tanin, for your great job at the beginning, and thank you to Mikael and Renata, which are in the backstage, and they were taking care of comments, and to all the technical side of this live event. As always, we ask you to follow us on Facebook if you want to stay in contact with us for everything. And I will also suggest you to subscribe to our YouTube channel and Instagram and Twitter account. And we are coming back with another Couch of Science event probably in November and probably also in December. So you can stay in touch. If you want to see this live again, because maybe you reach us a bit later, you can find on Facebook and on our YouTube channel. You can find also all the other talk that we had with brilliant scientists like tonight. So please continue to follow us, spread the word about these events, because the more we are, the better. And Professor, thank you again. It was really nice.
Thank you. Thank you, Michele. Thank you, Tanin. And it was really a great pleasure. And good luck for the next Couch of Science. And please continue this type of initiative. It's really wonderful. Thank you.
Thank you so much. Thank you so much.
Thank you for your contribution.